So as we've just talked about, ATP production and the presence of ATP as a high energy carrier is really important for the cell. So now we're going to look more closely at how we make ATP in the electron transport chain. So let's first back up and ask what the role of the electron transport chain. And really that starts with the foods we eat, our major macromolecules, fat, carbohydrates, and protein. And as we'll see in the upcoming weeks, we oxidize these compounds into smaller molecules that are then used to generate reducing equivalents, shown here as 2H. And we then collect these reducing equivalents, and they're oxidized in the electron transport chain to ultimately produce ATP. What this slide really helps illustrate, and what we'll talk about later, is the interconnection between the ETC, the citric acid cycle, and all of our upstream metabolic pathways. So our setup of the mitochondria is illustrated here in this double membrane structure with an outer membrane, which is permeable to a lot of compounds, but we'll also see it's impermeable to NADH and FADH2, and this is important and the reason why we have our shuttle systems we'll talk about in a little bit. Then we have this inner membrane, which includes our electron transport enzymes as well as ATP synthase. Within the mitochondrial matrix, this is where we have our citric acid cycle enzymes, our enzymes for beta oxidation as well as pyruvate dehydrogenase, which is really our interface between glycolysis and mitochondrial metabolism. The other thing I just want to refresh everyone's memory with from general chemistry is the concepts of oxidation and reduction. So again, oxidation and reduction are the coupling of the loss and gain of electrons. And we'll be talking about this a lot in the course. So oxidation, or to be oxidized, means to lose electrons. Don't confuse this with the term oxidizing agent. If something is an oxidizing agent, that compound itself is reduced in a reaction. Just for simplicity, I have NAD+ here, and this is the oxidized form of NADH. So the term reduction or reduced refers to the gain of electrons. Again, don't be confused with the term reducing agents, as these are compounds that are oxidized in a reaction. And NADH here is the reduced form of NAD+. So be sure you're clear on these two terms, as we'll be using them quite a bit. So here we have a schematic of the components of our respiratory chain complexes, and we should keep in mind that the respiratory chain collects and transports reducing equivalents, directing them to our final electron acceptor here, which is oxygen. These complexes sit on the inner mitochondrial membrane, and complexes 1, 3, and 4 cross the mitochondrial membrane, whereas complex 2 does not span the mitochondrial membrane. So you'll notice here it's not pumping protons. And complex 2 is also part of the TCA cycle as succinate dehydrogenase. Other important things to note is that both Q, or also termed ubiquinone, as well as cytochrome C, are the only mobile parts of this electron transport process. All of the other complexes are seated in the mitochondrial membrane and are not mobile. So if we look at the flow of electrons, starting here, the predominant flow comes in as NADH. So here you can see electrons from NADH are transferred first to complex 1 through a flavin mononucleotide, and then through an iron-sulfur center containing complex. The same is true here for complex 2, where the electrons from FADH are transferred to FAD through an iron-sulfur complex, and then again to ubiquinone. From here, ubiquinone will transfer its electrons to both cytochrome B and cytochrome C in what's referred to as the Q cycle, and we'll look at that on the next slide. Ultimately, cytochrome C will donate 4 electrons to our heme complex within complex 4. Ultimately, the electrons from complex 4 are passed on to molecular oxygen until it's fully reduced to form water. So now let's look quickly at the Q cycle. And if we remember, Q is one of our mobile components of the respiratory chain. It can be found in 3 different forms. The fully reduced form, or quinol, is denoted here as QH2. This molecule carries 2 protons and 2 electrons. You have a fully oxidized quinone form, or Q, which has lost both electrons and both protons. And then the semiquinone, which is a free radical that's formed only transiently in this cycle. So as mentioned previously, Q is able to donate its electrons to both cytochrome B as well as cytochrome C. As the reduced quinol carries 2 electrons and 2 protons, we form this semiquinone during one of these cycles, as the cytochromes can only accept 1 electron at a time. So as the electrons are donated to our cytochromes, this is coupled with the movement of protons across the inner mitochondrial membrane into the intermembrane space. So we've looked at this first part of the electron transport chain up to the formation of water. What we haven't addressed is the use of our protons on the intermembrane space. And the flow of protons back across the inner mitochondrial membrane through complex 5, or the ATP synthase, is what allows us to then use this electrochemical gradient to generate ATP. So the ATP synthase is drawn here, and it consists of this F0 group, which contains these C subunits which sit within the inner mitochondrial membrane. This is attached to this gamma subunit, which functions more or less as an axle or a spindle as it runs through the middle of this protein head group. This head group is the F1 head group, and it consists of alpha and beta subunits. So what's important to note is these alpha and beta subunits are fixed, and these do not rotate as part of this process. So each subunit will uptake a ADP and a free phosphate, and the movement is really within this F0 component as protons flow through the F0 subunit. This causes a shift and allows this bottom piece to turn, as well as this gamma subunit to turn, causing a catalytic change which results in the production of ATP. So let's finish with a clinical correlate, and we'll revisit this clinical correlate later when we talk about anaerobic metabolism. So there's a heritable condition termed MELAS, or mitochondrial encephalopathy, lactic acidosis, and stroke. And this is caused by a deficiency in either complex 1 or complex 4 in the electron transport chain. So let's ask the question as, how does this lead to lactic acidosis? And what I want you to appreciate is that the electron transport chain is the fundamental end of metabolic pathways. And dysfunction in the electron transport chain can cause a host of upstream problems, such as lactic acidosis. So globally, glycolysis will oxidize glucose. The glucose oxidized to pyruvate would naturally flux into the TCA cycle, and we'll talk about that in a couple weeks. And those reducing equivalents produced by the oxidation of glucose and subsequent acetyl-CoA in the TCA cycle should be oxidized through the electron transport chain, as we've just talked about. So again, we have glucose to pyruvate. Pyruvate oxidized through the TCA cycle to produce NADH and FADH2. And what we've just seen is that these reducing equivalents can be oxidized through the ETC to make ATP. And we're asking the question as to what happens if this is dysfunctional. So what happens is a series of feedback inhibitions that result in the accumulation of pyruvate. So we'll see later that high levels of NADH will negatively inhibit the TCA cycle. And with reduced activity of the TCA cycle, you'll have accumulation of pyruvate. Pyruvate will then be shunted to produce lactate through the reaction of lactate dehydrogenase. So what I want you to appreciate is that dysfunction far downstream of metabolic pathways has upstream clinical consequences.